Bacterial metabolism with diagnostic correlation. Metabolism is the sum of all of the chemical reactions in a cell. With bacteria, just like people, they gotta eat. And bacteria are going to be able to consume different types of metabolites based on what enzymes they have encoded in their genetic material. So there's three major macronutrients, or three ways that bacteria can get nutrients from their environment. That's by consuming carbohydrates. Carbohydrates are sugars, lipids, which are fats, and proteins. Carbohydrates, those sugars, come in different varieties. So monosaccharides contain one building block, disaccharides contain two building blocks, polysaccharides contain many building blocks, and bacteria have different enzymes to be able to break down those substances into smaller substances, and then they can absorb those into their cell, and then internally they can digest them through many different metabolic processes. Lipids are pretty big, so in order for a bacterium to be able to eat a lipid, it definitely has to release lipid digesting enzymes into the environment so that it can break down those lipids into more manageable pieces or building blocks so that it can absorb those building blocks. Proteins are also pretty large, so there's proteolytic enzymes that'll break down the proteins into their amino acids, and then the bacteria can bring the amino acids in. But even once they're in, those substances, those building blocks, have to be metabolized further. And there's different processes of how bacteria can do this. Why does all of this matter? Why do we care what bacteria eat or what they do in order to metabolize different substances? Well, when we first started discovering the differences of bacteria, we started looking at them under the microscope, and we're like, well, some bacteria are circular and some are rod-shaped, and some look like this and this and this under the microscope, and we can do these different staining techniques to help differentiate them. But then we discovered that there's hundreds and then thousands and now millions of different species of bacteria, and we're continuously identifying new species of bacteria out there. So we can't just look at them under a microscope or stain them a certain way to be able to identify them. Now that'll give us some good tips and clues, especially with certain infections in certain regions of the body, what the bacterium is, but we have to take it a step further. Back when the field of microbiology was exploding, we didn't have a lot of DNA technology, so it wasn't easy to just do a whole genome sequence of a bacterium or to be able to sequence a particular part of the bacterial DNA in order to identify it. So how would we identify bacteria? Well, we can put the bacteria in a tube with some sugar and a pH indicator. And if that bacterium has the ability to eat that sugar, it'll eat it and then produce an acid waste product. And that gets detected by that pH indicator. So we started figuring out, well, if we put the bacteria in a tube with select types of nutrients, and if the bacteria can eat it, it's going to cause a pH change, which is going to cause that indicator to turn colors. That's how we can start to list characteristics of the different microorganisms. And if we know that a species of bacteria has the capability of eating select nutrients, we can compare our unknown to what we know. And if it has the same metabolic pattern and the same cell shape and the same staining characteristics, we can give an identification of that bacteria. To this day, we still use those metabolic tests in order to differentiate. Now today, they look a lot different. They used to be these giant tubes, and we used to inoculate the bacteria in all these different tubes and get an arrangement, and then we made them into a smaller and simpler system where we could batch 12 tests in a very small area. And then from there, we made it an even simpler system where now we have automated machines that can detect color changes. So as soon as a bacterium eats something, it can detect that color change and it can report out the arrangement or the metabolic characteristics the arrangement of tests that would turn positive, and compare that to a bunch of known species, and we can get an identification that way. We also have a lot more technology, which we'll talk about as we go through the next couple lectures here, but I just wanted to give you this background of why you should care about this stuff. You will have to know the details of many of these tests as we go throughout the course. So as I mentioned previously, you want to make sure to focus on what's in the study guide and don't try to over-memorize. There's a lot of content provided here. Only worry about the content that's in that study guide, and we will start learning as we start actually performing these tests in the lab. Then you're going to have to start to know all of the details of each one of these metabolic tests. So as a review, respiration or cellular respiration can be aerobic or anaerobic. In aerobic respiration, it's very efficient. There's energy generating process in which molecular oxygen is the final electron acceptor. Anaerobic respiration is similar. They go through all the same steps, the glycolysis, Krebs cycle electron transport chain, except something other than oxygen is the final electron acceptor. Fermentation is an anaerobic process that's carried out by both obligate and facultative and aerotolerant anaerobes. Electron acceptor is an organic compound, and it's less efficient in energy generation than respiration. So bacteria, different species, have the ability to do different processes. Even within one species, they may be able to do multiple processes. So we have to have a basic understanding of all of these processes in order to understand how bacteria eat or metabolize certain nutrients. So if the bacterium does do fermentation, so fermentation is basically glycolysis, and then pyruvate gets, at the end of glycolysis, gets converted to a variety of different substances. Here are the fermentation pathways that we know of. So there's alcoholic, homolactic, heterolactic, propionic, mixed acid, butanediol, and butyric acid fermentation. So again, glucose gets brought into the cell. Glucose is broken down through the process of glycolysis. It's a 10-step process that ends in pyruvate, or 2 molecules of pyruvate. Pyruvate can then be further metabolized. So pyruvate or pyruvic acid can be converted to acetyl-CoA and then ethanol and acetic acid, acetylactic acid, acetone, and 2,3-butanediol. Oxaloacetic acid, succinic acid, and propionic acid. It can go to acetaldehyde and ethanol. It can be converted to lactic acid, formic acid, acetoacetyl-CoA, which can then be broken down into acetone and isopropanol, and butyryl and CoA and butyric acid and butanol. Alcoholic fermentation is used by yeast to ferment glucose to produce ethanol. Very few bacteria have the ability to produce alcohol from fermentation. So we don't usually focus a lot on alcoholic fermentation, and our metabolic tests are not designed around the detection of alcohol fermentation. Homolactic fermentation is used by Streptococcus and Lactobacillus. The end product is lactic acid. If you think about producing yogurt, so you get milk, you throw some lactobacillus in there, and that lactobacillus is going to consume the products within the milk and produce lactic acid. And that lactic acid makes the proteins clump, and that clumping of the protein, that's what you see with the yogurt. Heterolactic fermentation is used by some lactobacilli. End products are lactic acid, carbon dioxide, alcohols, formic acid, and acetic acid. Propionic fermentation is used by select gram-positive bacteria. The end product is propionic acid. Mixed acid fermentation. It's used by select gram-negative rods in the Enterobacteriaceae family. Carbohydrate fermentation with an end product of lactic, acetic, succine, and formic acid. So it produces a mixture of acids at the end. Butanediol fermentation is used by select gram-negative rods in the Enterobacteriaceae, and it's based on carbohydrate fermentation. Butyric acid fermentation, obligate anaerobes use this pathway to produce butyric acid, acetic acid, carbon dioxide, and hydrogen. The aerobic utilization of pyruvic acid, or oxidation. The Krebs cycle, pyruvic acid is oxidized, energy in the form of ATP is generated, and the cycle results in the production of acid and the evolution of carbon dioxide during aerobic respiration. So once the glycolysis is complete, if fermentation doesn't happen, and if there's oxygen present, we can use it in the aerobic cycle of Krebs cycle. So historically, as I mentioned, biochemical testing was based on phenotypic characteristics. And then we had serotyping, which uses antibodies to detect specific antigens located on the bacterial surface. We will talk a lot more about the antibodies and serotyping when we get to the immunological diagnostics lecture. Newer technology is available. The matrix-assisted laser desorption ionization time-of-flight mass spectrometry, or MALDI-TOF MS, is based on the characterization of microbial proteins. So we put a small sample of isolated bacteria onto a tiny dot on a metal plate. We put that metal plate into the MALDI-TOF. A laser hits it and breaks apart the cells and causes the proteins to then take flight. So the proteins are lifted up off of the sample and they go through the mass spectrometry detector, which detects the different proteins and different characteristics of those proteins, producing a characteristic graph. And when that graph is produced, it's compared to a database of many different graphs of known species, and then we can determine the species or identity of the microorganism based on the graph or the arrangement of proteins that was detected by the MALDI-TOF. Molecular biology is based on genotype. Based on nucleic acid sequences, it's highly sensitive, specific, and fast, providing accurate results in a few hours or less. Molecular biology is a growing field. A lot of people are speculating that pretty soon there will not be a microbiology section in the lab. It will be a molecular biology section. So traditional tests for gram-negative bacteria: triple sugar iron, methyl red, and Voges-Proskauer test, the indole test, urease, Simmons citrate, carbohydrate fermentation, lysine iron agar, and sulfide indole motility, or SIM. These tests will be performed throughout this course, so it's kind of a good idea to take some notes about what these tests are, and we'll talk more about them as we go through these next few slides. So starting with carbohydrate utilization. So as I mentioned, bacteria are going to consume macronutrients. And when bacteria consume those, we've got the carbohydrates, the lipids, and the proteins. So we're going to start with carbohydrate utilization because different bacteria can eat different types of sugars. So lactose utilization is probably one of the most important tests. This helps differentiate the enterics. So enterics are the bacteria that live in the gut. So if a person comes in with diarrhea due to bacterial-related infection, and we want to culture the bacteria and figure out what's causing the issue, lactose fermentation is a really good way to differentiate those gram-negative bacteria. So it's used to differentiate the lactose fermenting LF bacteria from the non-lactose fermenters NLF. Lactose is a disaccharide bond consisting of glucose and galactose connected by a galactoside bond. Lactose degradation requires two enzymes, beta-galactoside permease, which transports the lactose through the cell wall, and beta-galactosidase, which breaks lactose to yield subunits. Some bacterial species lack permease but have the beta-galactosidase, and those are known as the late or delayed lactose fermenters. After lactose degradation, glucose is available for metabolism. Oxidation fermentation tests. So in some cases, we want to know whether a bacterium has the ability to use a certain carbohydrate either in the presence of oxygen or without oxygen. So in the presence of oxygen, we would call that the oxidation test. Without oxygen, we would call that the fermentation test. So we usually have two tubes next to each other. One of them we allow to be exposed to oxygen. The other one, we pour some mineral oil or some type of oil on top of it to create an anoxic environment, so an environment without oxygen. And then we put the bacteria in each of them and see what the bacteria can do. Can it use a particular carbohydrate in the presence of oxygen or not? And then can it use a particular carbohydrate in the absence of oxygen or not? And we can get a pattern of that, and then that's more data that we can then confirm to known results. So oxidation uses it aerobically, fermentation uses it anaerobically, and asaccharolytic means that they do not utilize carbohydrates. Anytime you put the letter A in front of something, it usually means that it's the absence of. So A, the absence of, saccharolytic. We're talking about sugar. Lytic is the ability to break apart sugars. So these asaccharolytic microorganisms do not have the ability to break apart sugars. Learning these stems and being able to look at a word and breaking it down into its stem is really a good study technique. So as we go through this course, if I see any terms that lend themselves well to being broken apart, I will call those out and kind of break them down for you. So fermentation, the glycolysis pathway, glucose to pyruvate, and then it's oxidized to other acids to products and gases. Acid is then detected by pH indicators. There's single acid fermenters and mixed acid fermenters. Oxidation is the glycolysis pathway. Glucose is converted to pyruvate, and then the aerobic pathway requires oxygen, or the anaerobic pathway requires oxygen-containing an inorganic molecule. Generally weak acid producers, the acid is neutralized by alkaline reactions from the peptones. So OF basal medium, OFBM, is a lower concentration of peptones. It helps classify an oxidizer or fermenter. It detects small amounts of acids. A pH indicator is bromothiamyl blue. Uninoculated is green. Acid pH is yellow. Alkaline pH is blue. Uses two tubes, as I mentioned. There's an aerobic tube, which is an open tube. The anaerobic tube is overlaid with sterile mineral oil. The anaerobic is only closed tube, produces acid. Facultative, both tubes produce acid. So if we see an obligate anaerobe, it would only be able to grow in the presence of that mineral oil or in the safety net of that mineral oil preventing oxygen entry. If it's a facultative, then it would be able to grow in the aerobic tube and the anaerobic tube. Non-oxidizers produce an alkaline pH that appears blue in both tubes. So here you can see some sample OF media reactions. As I mentioned, here's the two tubes right here. So this is 1A and 1B. This means that this is the same species. This doesn't have mineral oil on the top, so this one's open. This would give us our aerobic result. This one here does have mineral oil that prevents oxygen entry into the tube, so this would give us our anaerobic result. Green is negative, yellow is positive. So we put a particular type of sugar in here, and then we cover up one with mineral oil to create an anaerobic environment. And it turned yellow, so both of these are positive. So that would mean that this is a facultative anaerobe. It means that it can ferment in the presence of oxygen or survive in the presence of oxygen. It can go through oxidative processes, so maybe aerobic respiration. And it can also go through the fermentation in the absence of oxygen, so it can produce that color change of the media. It produces acid, which causes the pH indicator to turn colors. In this next set, we see that the aerobe is positive and the anaerobe is negative. So it is an obligate aerobe. It cannot undergo the process of fermentation in the absence of oxygen. And this one may be asaccharidic. Maybe it just doesn't, it's not a non-oxidizer. It's not able to utilize that type of sugar, because both of these are negative. Triple sugar iron agar. It contains sucrose, glucose, and lactose. Lactose and sucrose are present in a 10-to-1 ratio to glucose. Klinger iron agar is similar but no sucrose. Ferrous sulfate and sodium thiosulfate detect the production of hydrogen sulfide, H2S, which is a black color. And then there's a phenol red pH indicator. If it's an acid, it's yellow. An alkaline reaction would be red, or base would be red. Reaction chambers, the slant is aerobic because it's exposed to oxygen. The butt, or the bottom part of the slant, is anaerobic. So before we go on, I just want to show you what is meant by a slant here. So this is the slant. So the bottom is an anaerobic environment, and the top is an aerobic environment. So it must be read at 18 to 24 hours. It's read as the slant over the butt. If there's no fermentation, it would be alkaline over alkaline, or K over K. No lactose or sucrose fermentation only. Glucose fermentation only initially first 12 hours. Acid A over A then converts to K over A. K over A is typical of glucose fermenters. After glucose is used up, the peptones in the slant get degraded, leading to a higher pH. Anaerobic fermentation produces more acid in the butt, therefore it stays yellow. Lactose or sucrose or both fermentation. You see A over A. After using glucose, then the lactose or sucrose is used, so both stay yellow. Hydrogen sulfide production, black precipitate. Acid from bacteria and sodium thiosulfate, H2S gas plus ferric ions, equals ferrous sulfate, which is a black precipitate. Gas production, you're going to see bubbles or the splitting of the media due to gas generated, and a lot of times that gas is carbon dioxide gas. So we predominantly use the TSI agar. The carbohydrates are glucose, lactose, and sucrose. There's an acid butt, acid or alkaline slant, alkaline butt, alkaline slant, alkaline butt, alkaline slant. So you can see the fermenter, what it would look like, and the non-fermenter, a non-oxidizer. So here's a sample of different TSI reactions. So in this first one, you see it's yellow on the top and yellow on the bottom. So yellow means that an acid was produced in both environments. This would be reported as A over A, acid over acid. We would also report the presence of gas. You can see that there was some separation down here at the bottom, so this would be gas positive. In this one, this would be A over A, and it's black on the bottom because of that black ferrous precipitate, so ferrous sulfide precipitate. So this would be A over A, H2S positive. This next one, it's a little tricky to tell, but it kind of looks yellow at the bottom, and it's definitely red at the top. So this would be K over A, so the alkaline over the acid. This one here, this would be K over A, H2S positive. So it's got red on the top, black on the bottom, K over A, and then H2S positive and gas positive. You would see that there's a gas bubble here. And this last one would be K over K. So this is asaccharidic. It doesn't use those carbohydrates in the medium. So the purpose of this test is to determine if an organism is found to be a DLF, or a delayed lactose fermenter, one that lacks the enzyme beta-galactoside permease, but possesses beta-galactosidase, and is a true NLF. ONPG is structurally similar to lactose, but ONPG is more readily transported through the bacterial plasma membrane. So ONPG is ortho-nitrophenol beta D-galactopyranoside or side test. ONPG test, steps to perform it: you make a heavy suspension of bacteria in sterile saline, add 1 ONPG disc or tablet, incubate at 35 degrees Celsius, and positive results are usually seen within 6 hours. Beta-galactosidase hydrolyzes ONPG into galactose and O-nitrophenol, which produces a yellow color. Non-lactose fermenters remain clear. Yellow pigment-producing bacteria should not be tested because of potential false positive results. So here's examples of the ONPG test. So this would be a positive with the yellow color change, and this is negative. Glucose metabolism and its metabolic products. So glucose metabolism occurs in a variety of different pathways. They are referencing the Embden-Meyerhof pathway. It produces several intermediate byproducts, including pyruvic acid. Further degradation of pyruvic acid can produce mixed acids as end products. Enterics, so enteric pathogens that live in the digestive tract, take 2 separate pathways: mixed acid fermentation or butylene glycol pathway. The methyl red MR and Voges-Proskauer VP test detect end products of glucose fermentation. So you inoculate glucose-containing broth and incubate it for 3 to 5 days. You transfer half of the bacterial suspension into a clean, dry tube for the VP test. The remaining half is used for the MR test. MR negative cultures remain yellow after the addition of the pH indicator. So you're going to add methyl red, which is a pH indicator, directly to one of the tubes. And if it remains yellow and doesn't change color, then it's negative. If after you add that it turns red, then it's positive. Glucose to pyruvic acid, mixed acid fermentation, pH 4.4. A red color with the MR is a positive test. After the bacterial suspension incubation, add alpha-naphthol, which serves as a catalyst or color intensifier, and then you want to add 40% potassium hydroxide or sodium hydroxide. In the lab, potassium hydroxide is more often used. Then you want to gently shake the tube to increase oxygenation. VP test: if glucose is metabolized by the butylene glycol pathway, then you get a positive VP test. It produces butylene glycol and acetone, which is further converted to diacetyl plus potassium hydroxide plus alpha-naphthol gives you a red complex. So after you add those 2 substances in here, you're going to see a red color formation if it's positive. So examples of MRVP tests. Here you can see a positive result and a negative result, and a positive result and a negative result. So in this instance, red is positive, negative would be clear or remain yellow. Amino acid utilization. So amino acids consist of an R group, which is the group that makes that amino acid unique. Then every amino acid has the base to it. So it's a carboxyl group and an amine group. Bacteria can either rip off that carboxyl group and eat that, or it can rip off the amine group and eat that. So decarboxylase talks about removing that carboxyl group. So this tests the presence of the enzyme capable of removing the carboxyl group. They're specific for amino acids. So there's lysine decarboxylase, ornithine decarboxylase, arginine decarboxylase, and so forth. Dihydrolase, arginine to arginine dihydrolase, citrulline, ornithine, and putrescine. The molar decarboxylase base medium, broth medium used to detect decarboxylation, It contains glucose and peptones. pH indicators bromocresol purple and cresol red. It's specific amino acid at a concentration of 1%. You overlay it with oil. Uninoculated is purple, and initial fermentation drops pH 5 to 5.5, which turns the medium yellow. Two conditions must be met for decarboxylation to occur. It has to have an acid pH, an anaerobic environment, and if the amino acid is broken down, amines cause an alkaline pH shift, causing a purple color, which is positive. If the color remains yellow, it's a negative test. An LIA slant contains lysine, glucose, ferric ammonium citrate, and sodium thiosulfate. Deamination or decarboxylation of lysine is the primary function of the test. If deamination happens, it produces a reddish color with a yellow butt because of glucose fermentation. If decarboxylation happens, it remains purple. It can also detect H2S production. So here you can see various results of these slants. So it can either be all purple, and up here, that means decarboxylation. It can be part purple and part yellow with that black color. Black means it's H2S positive, and it would be deamination. So red on the top, yellow on the bottom. Here you can see red on the top and all black on the bottom, so this would be kind of similar to that one. And here you can see just the deamination happening, red top, yellow on the bottom. This is useful in the differentiation of Proteus, Morganella, and Providencia. So here is phenylalanine deamination. So phenylalanine deamination, what would happen is we have phenylalanine-rich medium, and if the bacterium has the ability to deaminate phenylalanine, we would put ferric chloride on here and see a green color change if it's positive, and it would not be green. It would turn this yellow color if it was negative. Then there's some miscellaneous tests that we can perform. So citrate utilization, also known as the Simmons citrate test. Citrate test determines whether an organism can use sodium citrate as a sole carbon source. The use of citrate results in an alkaline pH. This changes the indicator from a green to a blue color, and blue is a positive test. The inoculum must be light. Dead organisms can be a source of carbon, so you do not want to over-inoculate the slant. This is really hard to distinguish, but you can see that this is green, this is blue, and this is green. So blue would be positive. Deoxyribonuclease, DNase. Endonuclease is produced by bacteria that can break phosphodiester bonds. Streak organisms with a heavy inoculum on DNase plate. Incubate for 28 to 24, it should probably say 18 to 24 hours. Let me just change that real quick. Add 1 normal of HCl. 1 normal of HCl is exactly 1 molar HCl to the surface of the plate. DNA precipitates, but if DNase, then you see a clear halo of growth around the organism. Indole production. So indole is a degradation product of tryptophan amino acid degradation. So if the organism possesses tryptophanase, it can deaminate tryptophan, resulting in indole. Bacteria are inoculated into tryptophan or peptone broth and incubated for 48 hours before the indole testing can be done. There's the Ehrlich's indole test. Indole is extracted by xylene. Add xylene, shake the tube well. The Ehrlich's reagent, positive color would be red. Kovacs, add 5 drops of Kovacs reagent to the culture. A positive is red color. I've seen more often the Kovacs reagent used. I've also seen the DMA-CA indole rapid test being produced in clinical labs. So here you can see a positive versus a negative result. So the top of the tube is red versus a yellow or a clear color. Motility is determined by the microscopic examination of bacteria or observing growth in semi-solid medium. The ability of a microorganism to swim or move is another distinguishing characteristic. So just like looking at cell shape, we can also look at them under the microscope and see if they're swimming. Now this is really tough, and there's a procedure called a hanging drop slide where you put a drop of medium at the top of a cover slip and then hang it over a well that's in the slide and put it on the microscope, and you try to watch them swimming. But sometimes bacteria don't want to move, or they're not moving at that moment. So if you happen to look at them under the microscope, they may not be moving at that time, and that may be a false negative result. So we have an alternative test, the motility medium. Motility test medium has concentrations of 0.4% or less to allow for free spread of motile bacteria. A single stab is made at the center of the tubed medium. After incubation, movement away from the stab line or a hazy appearance throughout the medium indicates a motile organism. So that motility indole ornithine agar, it's a semi-solid used to detect motility and indole and ornithine decarboxylase production. It's useful in the differentiation of Klebsiella from Enterobacter and Serratia species. It's inoculated by making a straight stab down the center of the medium. Motility is shown by a cloudy or medium spreading growth from the inoculation line. Ornithine decarboxylation is indicated by a purple color throughout the medium, and indole is detected by adding Kovacs reagent. A pink to red color in the reagent area is a positive test. So here you can see motility agar results. So this is uninoculated. So if we stab into the center of this medium, this bacteria has the ability to swim outward away from the stab line. In this instance, it's not motile, so it just grew in the stab line. So motile, not motile. Nitrate and nitrite reduction. Reduction of nitrate to nitrite. Add N,N-dimethyl alpha naphthylamine, sulfonylate acid, and examine for a red color. Then you would know that nitrite is present. Nitrate to nitrite plus sulfonylate acid plus NNDN, the diazo red dye. Test verification, add zinc. If the red color remains, then nitrate was not reduced. We will be performing the nitrate reduction test, and you'll learn all about this and have to know the details once we get to that portion of this course. Oxidase description, so it's helpful in differentiating between the Enterobacteriaceae, which are mostly oxidase negative, and the Neisseria species. Modified oxidase is used to distinguish staph from Micrococcus. So the Kovacs oxidase test, several methods are available. Kovacs oxidase test uses 0.5 or 1% aqueous solution of tetramethyl P phenylethanol diamine dihydrochloride. Wow, that was a lot to say. Procedural highlights. Add a drop of reagent to the filter paper. Use a wooden applicator stick to rub bacteria colony into the moistened filter paper. And development of a lavender color within 10 to 15 seconds constitutes a positive result. Urease production. So urea is broken down by the enzyme urease. It's used to determine whether a microorganism can hydrolyze urea, resulting in a sufficient amount of ammonia produced to produce a color change by a pH indicator. Urease hydrolyzes urea to form ammonia, water, and carbon dioxide. The surface of the agar slant is inoculated. Phenol red is in the medium to serve as the pH indicator. The resulting alkaline pH from the hydrolysis of urea is indicated by a bright pink color. So here you can see this is negative and this is positive. Manual multi-test systems. So there are commercial systems that are available now. In these previous slides, most of the tubes were large like this. In these commercialized systems, the tubes are super small. And we have multiple wells with different reagents in them, and when we inoculate all those wells with the bacterium, we can see color changes in each one of those wells. And we report those color changes as being positive or negative, which comes up with kind of a numerical code system that we can then go look up in a reference manual. There are pH-based reactions, enzyme-based reactions, the utilization of carbon sources, visual detection of bacterial growth, or detection of volatile fatty acids. A number of packaged kit systems by which organisms are identified. There's computer-assisted databases and computer-derived numerical codes. Metabolic profiles are determined based on the biochemical reaction results. So plus for positive, and then we see the negative sign, and sometimes you'll see the equals sign for a negative result. The equals sign is used for negative because sometimes a dash is missed. So a positive is really easy to identify. If we have a negative result, you might put that equals sign. So the API test system. API is a system for the identification of gram-negative fermentative bacteria. API 20E means enterics. There's also a 20A for anaerobes, and there's a bunch of different API systems to detect different types of organisms. It consists of 20 couplets attached to a plastic strip, each with specific lyophilized pH-based substrate. A liquid bacterial suspension is used to rehydrate all the cupules. Let me just go to one of these systems. This is somewhat what it looks like. So each one of these cups contains a different type of medium, and then we put the bacteria in all of the wells, and then the bacteria are going to interact with the media to either cause a color change or not based on whether they are metabolizing that substrate. Some cupules require a mineral oil overlay. The principles of the test are the same for similar tests performed in those larger tubes. The strip is incubated for 18 to 24 hours, and then reagents are added as appropriate. Results are recorded, and a 7-digit code profile number is determined. The code profile number is checked against the database provided by the manufacturer for identification. Additional testing may be needed to distinguish different species, but for the most part, this is pretty solid at being able to identify. Rapid and automated identification systems. So this is increasingly important today. The term rapid is based on the time of the test versus the conventional method. So the conventional method would be the tube test. This means that it's a little faster than the tube test. Select commercially packaged kits or fully automated instruments are now available. So in commercial kits, they use chromogenic or fluorogenic substrates, and it's based on enzymes. Due to the amplification effect, substrates change color quickly. Some results may be reached after 2 to 6 hours of incubation. These are some of the rapid tests that are performed: the spot indole, ONPG, oxidase catalase, biosolubility, PYR, rapid urease, rapid hipurate hydrolysis, MUG, and LAP. We will be performing about 95% of these tests in this course. So definitely take note of this table. Take note of the mode of action, the applications, but you won't have to have all of these details memorized until we actually perform the tests in the lab. Automated identification systems. So most automated systems use turbidity, colorimetry, or fluorescent technology. These systems have a variety of capacities depending on laboratory workflow. Some examples are the microscan. This is a microscan. We have one of these. We do not have the walkaway. We have the smaller one. But basically you get a panel with all different types of substrates on there with color changing abilities, and you inoculate the bacteria on the entire panel, and then the computer system will read the color changes. This can also determine antibiotic susceptibility. So the computer system will, with all of the different wells, they're inoculated with different concentrations of different antibiotics. So in addition to testing whether the color changed in the media for the identification, it can also detect turbidity or the growth in the bacteria in the presence of different concentrations of antibiotics. The Trek diagnostic system, Vitek 2 is very similar to that system. The BD Phoenix automated microbiology system, Biolog, OmniLog ID system, and Sherlock microbial identification system. These are all different systems that you may encounter in practice. The two that I've seen the most in practice are the Vitek and Microscan. And here's an example of the Sherlock microbial identification system. So the evaluation of the identification system, you want to compare them side to side to the current methods of reference procedures. You want to test accuracy, cost effectiveness, and workflow. Systems can provide decreased sensitivity or specificity for some bacteria. Usually supplemental and differential media and biochemicals will still need to be kept on hand.